morning. My name is Amira Hanafi. I am a fresh lecturer at the Faculty of Pharmacy, Fars University in Egypt. I defended my thesis just last month, and it was about designing polymeric nanoparticles, biodegradable of course, to target the brain, mainly for Alzheimer's disease management. And during my thesis, one of the parts that I was researching was the toxicity of those nanoparticles, because in my humble opinion, I think that this area of research has not gained the attention it deserves up till now. So my talk today is entitled Polymeric Nanoparticles for CNS Targeting. It is time to think about the nanotoxicological aspects. The objective I am aiming at through this presentation is to redirect our interest as pharmaceutical researchers to think more about the nanotoxicological aspects. And to do this, we have to collaborate with the pharmacologists and the toxicologists in order to push forward the development of such nanoparticles and deliver them to the market. The polymeric nanoparticles that are usually used for CNS targeting include many examples. The most famous ones are the PLGA, chitosan, caprolactones, and many others. As pharmaceutical researchers, let's be honest. What is the main focus of our research when we deal with polymeric nanoparticles? Actually, we think about optimizing the formulation. We just focus on the formulation and its stability, maybe its efficiency, by collaborating with pharmacologists. For those who have a little bit industrial perspective, they will consider this one, the processability and the cost-benefit ratio. However, very few of us will think about the administration safety. And if we thought about this, it will come at last, not at first. And this is the main problem I am willing to discuss today. This figure is based on the ISI database checked on 2014. It shows here that this bar represents the number of articles between 2011 and 2013, which was relating to polymeric nanoparticles in general. And this small bar only refers to those articles who were referring to the toxicity of these polymeric nanoparticles. So the question is, however, those nanoparticles have been pioneered since 1999. The research is going for more than 15 years and even more. What about the clinical trials? Is any of our research have succeeded to reach this stage, to reach clinical trials? Up till now, up to this moment, there is none. Although, however, despite of all the effort we are making. Why is that? Many factors, of course, contribute to this failure. But one of these factors is the in vivo fate and the safety of such nanoparticles, because actually this era of research is still very vague. We do not know for sure what these nanoparticles will be doing, how they will be interacting with the tissues when we administer them, whatever the route is. So if we are talking about preparing polymeric nanoparticles, especially targeting the brain, what are the factors that we should consider which will affect its safety? First of all, the polymer's biodegradability. Of course, we cannot use anything but biodegradable polymers, like the ones I said before. However, we should not consider this factor only. We are making a big mistake if we thought that because the bulk material itself of the polymer is safe, that necessarily means that the polymeric nanoparticle resulting from this bulk material will be safe. This is a huge mistake and very common also. Why? Because actually converting a bulk material into polymeric nanoparticles completely changed the size and the surface properties of these nanoparticles. And those two factors may be the responsible ones for all the toxicological events happening inside the body. Also, we have to consider the surface coating we use in the formulation. We have to consider their tendency to aggregation, because this particular factor may lead to provoking some immunological responses in the body, and also the addition of stabilizers, which may be toxic themselves. This is a very interesting study that was performed in 2011. They investigated the toxicity of 200 nanometer chitosan nanoparticles on zebrafish embryos. They actually incubated these nanoparticles with zebrafish embryos, and then they observed the hatching rate, mortality, and any malformations that may have occurred after hatching. They found that those nanoparticles led to the formation of embryos with bent spines and edematic yolk sac. And this actually, in the opinion of the authors, of course, may be because of the apoptotic reactions initiated by the endocytosis of such nanoparticles into the embryos. So if we now decide to focus more on the toxicity of such nanoparticles, we have two ways to follow, either using in vitro models or in vivo models. The in vitro toxicity assays are basically the cell cultures. Very simply, we incubate the polymeric nanoparticle with the cell culture and measure some parameters. The cell viability parameters, like the morphology, any apoptotic reactions. We can also measure the lactate dehydrogenase and the MTT assays or tests, and we can also measure the inflammatory cytokines. However, these cell culture techniques appear to be very simple and straightforward, so they have their own merits and demerits. The advantages of such systems include they are fast, devoid of any ethical issues, easily automated, and of low cost, of course, compared to the in vivo models. However, we cannot rely on these systems and forget about the in vivo toxicological models. Why? Because there is many interfering factors, like selecting the cell type you use and the amount of the polymeric nanoparticles that you will decide to incubate with the cell culture. All these factors may lead to false positive or false negative results. Stability of cell lines, of course, which is a big problem. You need to establish first a correlation between your in vitro model and the in vivo reality before relying on such models. Also, there is no toxicokinetics. You cannot perform a toxicokinetic study using such cell culture models. And also, you cannot, of course, test for chronic effects of such polymeric nanoparticles. Therefore, alternatively, you have the in vivo toxicological methods. When we talk about the brain targeting, we usually use mice or rats. We can monitor many parameters, such as the animal's body weight, food intake, morbidity, mortality, blood biomarkers, and we can also test for the toxicity of those polymeric nanoparticles in certain organ, like the brain in our case, by performing histopathological examination and performing the same assays we said before. The advantages of such systems are, first of all, you can perform a localized organ toxicity evaluation, you can perform a toxicokinetic study, and you can study the chronic effect, of course, of such formulations. However, working with animals is generally very time-consuming and laborious. I tried it myself. Very expensive, and very full of ethical issues, of course. During my PhD thesis, I prepared something called galantamine chitosan complex nanoparticles. Actually, it is a complex of the galantamine, which is a well-known anti-Alzheimer drug, with the classic chitosan nanoparticles. But I had to make a complex first between them because of the low entrapment efficiency of galantamine into such nanoparticles. Anyhow, chitosan polymer itself has been tested for its toxicity in very numerous research which have been already published. However, there is no in vivo toxicity of chitosan nanoparticles for brain drug delivery up to this time. None. This is actually a hypothetical model I draw for the galantamine chitosan complexation, which I prepared. And that's why I had to study the toxicity of such nanoparticles. Why? Because, first of all, there is no studies up till now about these chitosan nanoparticles. And the second reason is that I prepared a complex which can be considered a new chemical entity in a way or another. So that's why I thought maybe it provokes some immunological responses or completely change the toxicological profile of the drug itself. I designed the study on healthy male Western rats for 12 consecutive days. I divided the rats into 4 groups: negative control group, oral galantamine solution group, nasal galantamine solution group, and the nasal galantamine chitosan complex nanoparticles treated group. The toxicological aspects or parameters that I was monitoring were the clinical signs, blood biomarkers, and I have performed histopathological examination in the brain at the end of the study. Regarding the body weight and food consumption, I found that there was a significant difference between the oral galantamine group and the negative control group regarding the body weight. There was incidences of diarrhea and weight loss. However, talking about the galantamine nanoparticles, they were insignificantly different from the control group. Regarding the food intake, the single group that showed significant difference was the oral galantamine treated group. There was a significant decrease of the food consumption at the end of the study. And actually, that's why I performed my study in the first place, because galantamine is well known for its severe side effects, which may lead, in some cases, to stopping or discontinuing the whole therapy. Regarding monitoring the blood biomarkers, I chose some parameters such as urea and creatinine as markers for kidney functions, aspartate and alanine transaminases as markers for liver functions, and differential white blood cells as marker for immunocompatibility. Regarding the urea and the serum, I found that there was no statistically significant change between the nanoparticles treated group and the control group in both urea and the creatinine serum levels at the end of the study. The same goes for the aspartate and the alanine transaminases. There was no significant changes. And the same applies for the total white blood cell count. There was no statistically significant change between the two groups. The last part was the histological examination. Actually, I made different sections in the brain of the rats, in the olfactory bulb, the orbitofrontal cortex, the hippocampus, and the parietal cortex. Upon examining those sections, I found that in the olfactory bulb, there was no histopathological manifestations in those sections between the negative control and the nasal galantamine nanoparticles treated group. The same goes for the olfactory, the orbitofrontal cortex, the parietal cortex, and also the hippocampus. One last thing, I thought about examining the orbitofrontal cortex sections specifically under the electron microscope at the end of the study. There was no ultrastructural abnormalities observed. And if I magnify these parts of the photo image, I will see here the nanoparticles, either inside vesicles or free in the cytosol. And that proves that those nanoparticles have been able to cross and be endocytosed inside the cells without affecting their viability. So my take-home message for you today is that as far as pharmaceutical researchers deciding to work on polymeric nanoparticles, we should not just focus on optimizing the formulation in vitro, or maybe extend our interest to the pharmacological efficiency and forget about the nanotoxicological aspects. I know there might be no time for questions, so I thought to give you my email address and my phone number, and if you'd like to discuss anything with me, you are very welcome. Thank you for your attention.
In fact, we still have time because a lot of people didn't show up. Very interesting talk, Amira, and it's an excellent. We'll start by the audience questions, and then I have one for you.
Yes, very interesting presentation.
Thank you.
Now, did you actually analyze or has any information or data that these nanoparticles actually reached brain?
Yes, I performed, I only showed a part of my study. I actually performed a pharmacokinetic study and a pharmacological efficiency study. I just focused on the toxicological part of my research because it was related to the topic. But I did this, yes, the nanoparticles reach the brain, and I have visualized them using fluorescence microscope, and I measured the pharmacokinetics, of course, and I also assessed their pharmacological efficiency, and they were far more pharmacologically efficient than the regular oral galantamine.
So how did you measure the amount of these nanoparticles in the brain? Is there a analytical method?
Using HPLC. Using HPLC.
So you
yes.
extracted the nanoparticles out of the brain?
Actually, I performed brain homogenization, and then I checked for the galantamine, the dissociated galantamine, using a fluorescence detector, HPLC fluorescence detector, because it's more sensitive than using the UV detectors. So however, one problem that I faced is to discriminate between the galantamine attached to the complex in the form of complex with chitosan or the free galantamine. Actually, the HPLC can differentiate between them, but the promising thing is that even when the drug is in the complex form, it proves to be efficient. Actually, I was very afraid that this complexation may cause any steric hindrance with the receptors or with the enzyme, actually, because it's acetylcholinesterase enzyme inhibitor. That's why I performed the pharmacological part, and it proves to be very successful.
That's very interesting, because you are right. Until now, they have not been concentrating on trying to figure out whether these nanoparticles themselves are toxic or not.
Yeah.
So good study.
Thank you.
Okay. Excellent. Okay, we have
thank you, Dr. Amira. Two questions. First, regarding study, there is no, any acceptance for a study without toxicological study. You know, even any new formulation or new chemical should be associated with toxicological study. Regarding chitosan, we have a huge work on that in the literature. I don't know if there is no toxicological study for this. I'm just asking.
Yeah, I am sure. Specifically, there is many toxicological studies that were performed on the chitosan solution itself using cell cultures and in vivo toxicity also in rabbits. In one study I saw. But there is no in vivo toxicity studies for chitosan nanoparticles designed to be delivered to the brain via intranasal route. This is the first of its kind. There is none.
You know, I last week or 2 weeks, I attended two conferences in Jordan.
Yeah?
In each one, we have chitosan nanoparticles, through nasal drug delivery.
Of course.
And
it's very common.
They have also studies on that.
Did they publish it? Did they publish it? Because up till now there is no publications.
One of the speakers there, she has many articles on that. She called Al-Ghananim.
I know them. I know this group of researchers. Actually, myself, when I searched for these articles, up to the moment when I decided to perform my study, there was none. Maybe in the past few months, something happened, and this actually makes me glad, because this era of research regarding the chitosan nanoparticles is very vague up till now, and this definitely makes me very glad.
Please, okay, one more question. Dr. Amira, is it okay?
Okay.
Okay.
Thank you.
It seems like a very interesting topic, like everybody wants to ask it.
Very, very interesting, and I think very relevant as well. Most of your points are well taken, and I do commend your research. I just wanted to know, would you say that the toxicity can be sort of exploited in a way if the nanoparticles reside in the brain as a form of toxicity? And what if the nanoparticles are actually needed in the brain? So that can be used as, it can be exploited to deal with some diseases in the brain, maybe?
Please repeat the last part of your question, because I didn't hear.
Okay, so I'm saying that if the brain has a condition which requires the presence of nanoparticles, let's say, can this toxicity that you have explained be used as a means to exploit? You know, I mean, it would be toxicity, but it's also relevant in the treatment of a disease that requires the nanoparticles to be present in the brain itself.
Yeah.
Can that be a positive in this negative?
Actually, if I understood your question properly, part of my research during the PhD was to assess something that is well known for those nanoparticles, that they are drug reservoirs. The surprising thing was that those images I showed to you using the electron microscope are actually captured 24 hours after the last dose. The laking part in my study was, unfortunately, I have not performed this part, but I should have, is to investigate if those nanoparticles will reside for more time in the brain, because actually that's maybe the reason behind their improved pharmacological efficacy compared to the oral galantamine. But I intend to perform this part in the future.
Please join me to thank Dr. Amira. Thank you very much.